And then admit everyone. Okay. Hi everyone, welcome to the WebEVTalk. So I'm going to take a screenshot for of the participants. So if you can turn on your video just for a minute or two, that would be awesome. Okay. All right. Yeah, might take another shot in the middle of the talk. So okay. Yeah, not during your talk, but after you talk. I don't want to disrupt you just to get a screenshot. Anyways, yeah. Now let's start. Wait a second. Is that all right? Yeah.
Looks good.
Okay, good. Yeah. Welcome everyone to the WebEVTalk series. It's a talk series for EV enthusiasts. And we had a good run last year. And this time we're starting at the USA Asia region. It's on the 9th February 2021. So if it's your first time to join WebEVTalk series, welcome. And we have alternate weekly. So this week is the USA and Asia region, and next week will be the Europe and Asia, and then go back to the USA region again. So today is the first session for the USA and Asia region, and it's my pleasure to welcome Nicole Noren Hooten. Nikki used an integrated interdisciplinary approach to study how oxidative stress, post-transcriptional mechanism, and other pathways contribute to health disparities, aging, and age-related diseases. Recently, she has been interested in utilizing extracellular vesicles as non-invasive biomarkers of aging and age-related diseases, and to further understand how various age-related diseases alter EV's cargo and function. She poses this question in the context of human aging through her work with the Healthy Aging in Neighborhoods of Diversity Across the Lifespans, or HANDS study. So today, Nikki is going to present her talk on extracellular vesicles and associated cargo in aging. Please welcome Nikki.
All right, thank you. Okay, so I'll go and share my screen. Does everything look all right, Carolina?
Just wait a minute until it goes away, that floating thing. Yeah, now it's good.
Okay. Okay, it went away. Okay. All right, first off, I'd like to thank Carolina and Alissa for moderating today, and also Carolina for and colleagues for organizing this seminar series. I've really enjoyed listening to the talks, and also I'm grateful for the opportunity to discuss our ongoing research projects. So aging is a complex, systematic process which is associated with the progressive loss of organ and tissue function over the lifetime, resulting in an increased risk of various age-associated diseases and mortality. In fact, aging is the biggest risk factor for most chronic diseases, including neurodegenerative diseases, cancer, type 2 diabetes, and heart disease. So how do we study the complex biology of aging? So I'm part of a basic science unit within the Healthy Aging in Neighborhoods of Diversity Across the Lifespan, or handle study. So we take a unique approach where we translate information from the bench to the neighborhood and vice versa. So handles is an interdisciplinary longitudinal epidemiological study of health disparities in aging and is based in Baltimore City, which is in Maryland in the United States. And it's been ongoing since 2004. It examines the effects of race and socioeconomic status on health outcomes in a cohort of urban adults. So at baseline, handles consisted of 3,720 community dwelling African Americans and whites aged 30 to 64 years. And what's unique about this study is that mobile medical research vehicles go out into the local Baltimore neighborhoods, and the MRVs provide a platform that enables recruitment and retention of non-traditional research participants into age-related clinical research. And you can find more information about the study here on the website or in this publication. So Handles tackles the problem of aging at the macro social level. However, the basic science unit within Handles studies aging at the organ, cellular, molecular, and genomic levels. And so our laboratory has had a longstanding interest in identifying non-invasive biomarkers of aging. And previously we examined microRNAs, which are small non-coding RNAs. And we found that microRNA expression decreases with age in peripheral blood mononuclear cells, and that specific serum microRNAs decrease with human age. And we explored the mechanism further and found that these changes were due to changes in the microRNA biomachinery that occur with age. We also examined another category of RNAs, including long non-coding RNAs, and found that lincRNA levels in PBMCs are altered with age and poverty. And most recently, we developed a comprehensive sequencing protocol and analysis pipeline to examine extracellular RNA, including small and large mRNAs in one sequencing reaction. And we did this as a function of age. And so here I'm not going to talk too much about these results because I don't have time today, but just wanted to highlight the distribution of extracellular RNA in young and old individuals. So here's a donut graph where each individual layer corresponds to extracellular RNA biotypes in one individual. And so you can kind of see visually in these donut graphs the differences between and distribution between young and old, and also the variability. And so we became interested in extracellular vesicles, since at this time when we were working on this project, it became certain that extracellular RNA in the circulation can be protected by being encapsulated in extracellular vesicles or bound to cholesterol or RNA binding proteins. Because I should mention the study here where we looked at extracellular RNA was only looking at extracellular RNA in total serum. And so we became interested in extracellular vesicles because they contain functional cargo. And so here I'm going to use the term extracellular vesicles with ICIF guidelines to encompass EVs that are derived from many different biogenesis pathways, including exosomes, apoptotic bodies, and microvesicles, since they overlap in size, density, and some cargo. So we've been interested in, can EVs be used as biomarkers for age-associated diseases? And so we've looked at EVs in the context of type 2 diabetes mellitus and have found that diabetic individuals have higher EV concentration and decreased levels of insulin signaling proteins, and also have increased inflammatory cargo, which elicits inflammatory signals in monocytes. And so here's one age-associated disease that we've looked at in further, and others have looked at various other age-related diseases and have found differences in EVs in cancer, cardiovascular disease, neurodegenerative diseases, and also other diseases. However, most of these studies have examined EVs secreted from cells in tissue culture or focus on specific disease versus matched control. So we wanted to take a different approach and to characterize EVs with human age. So do EVs change with age across the lifespan, over time? What about their concentration, size, and cargo? So we identified 74 individuals from the Handle study in 3 different age brackets. They were either young, 30 to 35, and this was 30 participants, middle-aged, which were 40 to 55, 30 participants, or old, 55 to 64, 14 participants. And this agent cohort was both African American and whites, and each of these groups were race and sex matched. And these participants had donated plasma at 2 different time points, so time 1 and time 2, and these time points were approximately 5 years apart. So this allowed us to have a repeated measures approach and to do both a cross-sectional analysis at time 1, a cross-sectional analysis at time 2, and a longitudinal analysis. So we have 74 individuals, so we isolated 148 isolated EVs from 148 samples, as shown here. And then we initially measured concentration and size using nanoparticle tracking analysis. So initially, we wanted to determine which EV isolation technique that we wanted to use for our study. And so we pooled plasma from a number of individuals and then aliquoted the plasma samples and then isolated EVs either using ultracentrifugation, exaquick, or size exclusion chromatography. And then we measured EV concentration using nanoparticle tracking analysis on a nanosite. And the concentrations are shown here. And then the numbers on top are the coefficient of variance. So at this time, when we started this study, we decided to use exaquick since it had the lowest coefficient of variance and allowed us to do more of a high throughput isolation of extracellular vesicles. So we isolated our vesicles and characterized them given MISA guidelines at the time. And so our EVs, as shown here, 3 young and 3 old, were enriched in EV markers flotilin 1, GSG 101, and CD9 compared to the EV free fractions. And our electron microscopy images show clear round vesicles as shown here in the size range of extracellular vesicles. We performed nanoparticle tracking analysis showing the diameter on the x-axis here and particles in the concentration here. And that these are a typical size distribution to EVs that are isolated from plasma. And again, we can examine size distribution at both visit 1 and visit 2. We looked hard, but we couldn't really identify any differences in EV size with age. However, if we look at the area under the curve and look at concentration using linear mixed model regression, we observed a decrease in EV concentration with age. And so age is on the axis here, and here's concentration. And again, we can perform 2 different cross-sectional analyses here. And in both of these analyses, we found that there were decreased EV concentration with age. So we also, given our approach, we can look longitudinally and look at EV changes over time. So this is a spaghetti plot where we have the young, middle-aged, and old, all in different colors here. And so the start of the line is the EV concentration at time 1, and the end of the line is the EV concentration at time 2, and the length of the line is time. So what this analysis showed us was that there was decreased EV concentration over time. And this experimental approach also allowed us to look at the similarities or differences between each individual and their EV concentration at visit 1 and compare it to the EV concentration at visit 2. And so if we compare each individual's concentration at visit 1 to visit 2, it's highly correlated. So this suggests that each individual person may have their own unique EV concentration profile. And so the decreased EV concentration with age that we observe could be due to 2 mechanisms, either a decreased release from cells or an increased internalization from cells. And so we decided to focus on this latter hypothesis. But in order to do this, we had to develop an assay. And so we developed a FACS-based assay to measure internalization of EVs by circulating cells. And so we isolated EVs from the different individuals across the lifespan. We labeled them with the fluorescent dye. And then we isolated PBMCs from a single individual. So these would be freshly isolated PBMCs from a human donor. And we concentrated on the 3 main cell types in the circulation: B cells, monocytes, and T cells. We then incubated our fluorescently labeled EVs from the different individuals with the PBMCs and performed FACS for PBMCs that were positive for extracellular vesicle internalization. And here are some of the markers that we used to identify the cells. For example, CD3 for T cells, CD19 for B cells, and CD11B and CD14 for monocytes. First, we had to actually examine what cell type in the circulation is the main cell type that internalizes EVs. And so here shown in the FACS plot here, it turns out these are monocytes. So here's the FACS plot. Here it is quantified over here with a number of different PBMC donors. And what we found was that the majority of EVs are taken up by monocytes, followed by B cells, but that the internalization by T cells was actually negligible. And this is important since T cells are actually the main cell type in your circulation, or I should say the most abundant cell type in your circulation. And we found that this assay is both time and dose dependent. And to test whether it's an active or passive process, we treated monocytes with latrunculin A, which is an endocytosis inhibitor. And so treatment with latrunculin A totally abolished EV internalization by monocytes. So this suggests in part that monocytes internalize EVs through endocytosis. So we wanted to get back to our original question: is the decreased EV concentration with age due to increased internalization by circulating cells? So to remind you, we had our EVs from individuals across a lifespan. We labeled them with PKH, then we incubated them with PBMCs from different donors. And then we did FACS to look at EVs that were internalized. And what we found was that with increasing EV donor age, we see increased B cell internalization or uptake. And here is a graph showing age and percentage B cell uptake. And these are 4 different PBMC donors, different individuals, so different individuals. And so we found a significant increase in EV internalization with human age of the EV donor. And again, this is consistent across the 4 different PBMC donors. And so just to summarize some of this part, what we found was that circulating EV concentration decreases with age, and that this may in fact be due to an increase in EV internalization by B cells. And just to summarize some of the data, because I don't have time to show you today, we found that EV donor age affects monocyte activation, but not internalization by monocytes. And so we hypothesized that proteins on the surface of extracellular vesicles may contribute to this differential internalization. And so we performed an EV array on the surface proteins in collaboration with Melina Jorgensen's group at Aalborg University, and also performed ELISA MSD assays on some cytosolic proteins. And again, this allowed us to look at proteins at both visit 1 and visit 2. So we performed linear mixed model regression analysis on both the longitudinal cross-sectional visit 1 and cross-sectional visit 2. And so I'm going to highlight the results that overlapped in all 3 of our different analyses, and that we found with age that the EV levels of CD151 and mucin 16, which is also known as CA125, were increased in EVs with age, and that apoptosis markers, cleaved PARP, cleaved caspase 3, and P53 were decreased with human age. So we're able to see age-dependent changes in EV protein levels. And so again, this approach allowed us to compare year 1 and year 5 with EV protein content. And what we found was that there's a significant Pearson correlation comparing visit 1 and visit 2 for 39 out of the 46 proteins. So here's like a little graphical representation of the data. So, because you probably don't want to see 39 different graphs. So this highlights the stability of each individual's EV protein profile from visit 1 to visit 2. And this is important because not many studies have looked at how does an individual person's EV protein or concentration profile change over time, especially a 5-year period. So just to summarize this part of my talk, we found that changes in EVs when concentration and protein content with human age, and that if we look at, if we compare a 5-year age difference, so compare visit 1 and visit 2, each individual has a relatively consistent level of circulating extracellular vesicles. And so this really shows the validity and accuracy of our measurements. And also is important because, as I mentioned, there are limited studies that have examined EVs over time from a single individual. And so since this study was published, we've continuously learned about extracellular vesicle cargo as summarized here in this graphic. And so we became interested in what other cargo may be changed in extracellular vesicles with human age. And so one of the hallmarks of aging is actually mitochondrial dysfunction. And so it's known that there's a decline in mitochondrial function, and this is an important contributor to the aging process and also for driving age-related disease. Mitochondrial DNA mutations drive premature aging in mouse models and increase with human age. And furthermore, mitochondrial copy number, so the number of mitochondrial molecules per cell, was also found to decline with human age in PBMCs. And so mitochondrial DNA can be released outside of the cell as what's known as circulating cell-free mitochondrial DNA. And this circulating cell-free mitochondrial DNA can be reliably measured in serum and plasma. It can act as a damage-associated molecular pattern molecule, or a DAMP, following cellular damage or stress. And circulating cell-free mitochondrial DNA have elevated levels in inflammatory diseases, some different types of cancer, not all, in association with trauma or tissue injury, such as after acute myocardial infarction or sepsis. However, most studies examine circulating cell-free mitochondrial DNA in whole plasma or serum. So we asked, are mitochondrial components, including circulating cell-free mitochondrial DNA, in extracellular vesicles? There's some data on this in the literature, and there's a really big interest recently on this field, and that mitochondrial components can be visualized in extracellular vesicles. So here's an electron microscopy image from EVs that were isolated from mesenchymal stem cells. And this is from this publication in Nature Communications in 2015. And I wanted to show this image in particular because I thought, given this virtual environment, you may be able to see it pretty good on your computer. And that there's a vesicle with cristae of a mitochondria that's inside the extracellular vesicle. This extracellular vesicle is pretty big, but there's been smaller vesicles with either portions or fractions or whole EVs that are in these other publications if you wanted to look at those. So mitochondrial components have been visualized in EVs. And circulating cell-free mitochondrial DNA can be detected in EVs from myoblasts, astrocytes, and glioblastoma cells grown in vitro. And circulating cell-free mitochondrial DNA is present in plasma EVs from women with hormonal therapy-resistant breast cancer. So is mitochondrial DNA present in plasma EVs under normal physiological conditions? And are mitochondrial components important functional cargo in EVs? So in order to test these ideas, here's our experimental design where we had plasma. We pulled plasma from 4 different individuals, so we mixed it. Then we aliquoted this plasma, and then we isolated EVs, and then the EVs, or EV depleted fraction. And we had this in 2 independent pools with 2 independent sets of individuals. We characterized the EVs according to MISIP guidelines, where we had performed nanoparticle tracking analysis, and this is a typical size distribution to plasma EVs. Here's our electron microscopy images showing clear round vesicles in the size range of EVs. And then we looked at markers that were enriched in our extracellular vesicles that are known vesicle markers such as CD81, flotilin-1, and here the negative marker GM130 is absent in our EVs but present in our cell lysate. And so then we developed a protocol in order to quantify mitochondrial DNA from extracellular vesicles. So we isolated the EVs using Exaquick. We DNase treated the EVs, and we DNase treated so that way to degrade any DNA that would be potentially on the outside of the extracellular vesicles, or also that co-precipitated in our isolation procedure. We then isolated the DNA and then performed qPCR. We also had the EV depleted fraction. We isolated the DNA and then performed qPCR as well. And we also took a slightly different experimental approach when looking at using qPCR for in our primer design, in that we wanted to look at, it's controversial in the field, whether circulating cell-free mitochondrial DNA is fragmented or if it's actually whole mitochondrial genome. So to get a better idea about the nature of the circulating cell-free mitochondrial DNA, we used four different primers as shown here. And that we call them based on the starting nucleotide. So this would be mitos 3164, 4625, 7878, and 8446. So we used all four of these primers in our qPCR analysis. And so we wanted to validate these primers. So we isolated the DNA again from plasma extracellular vesicles, DNase treated, and performed qPCR. And you can see a good amplification plot as shown here. And here's a positive control looking at helogenomic DNA. And then if we run these amplicons from here on a agarose gel and stain using CyberSafe theta bromide, we're able to look at and find a single amplicon as shown here in the size range that's expected given our primers. And so we can so these are validation that our experimental approach is working. And so what we found, so what we have here is that we have the pool from extracellular vesicles from the four different individuals, as I explained earlier. And then we have an independent set of extracellular of pooled plasma that we isolated EVs from. And then the EV depleted 1 corresponds to the blue EV1 and EV depleted 2 from the orange here. And what we found is that mitochondrial DNA is present in extracellular vesicles compared to the EV depleted fraction. And we see very similar results if we look at 316, all four of the different primers as shown here. So mitochondrial DNA is present in plasma extracellular vesicles. So is it inside the EVs or outside the EVs? And so we, in order to test this, we either treated the vesicles with DNase as we did previously, or we also had an EV-free, a mock-treated experiment to look at whether this EV is outside. And so you can see here in the green bars that there is lower levels of mitochondrial DNA when it is DNase treated. However, the difference between the DNase treated and non-DNase treated was not significant. So this just shows that mitochondrial DNA can be encapsulated in extracellular vesicles. So we wanted to also validate our results using a different EV isolation technique. So we isolated EVs using ultracentrifugation, as shown here in the orange bars, and the EV depleted fraction would be here in the red. And these were pooled plasma from the same individuals that we did exaquick or ultracentrifugation. And so here in these orange bars, you can clearly see that mitochondrial DNA is present in EVs that were isolated by exaquick here in the blue, and also by ultracentrifugation. And most recently, we also have confirmed that mitochondrial DNA is present in EVs isolated by a different technique, size exclusion chromatography. And so we found that, that mitochondrial DNA can be present in EVs isolated from a variety of techniques. And so we wanted to look at extracellular vesicles in our aging cohort. So we had EVs, and we had 55 EVs from 55 individuals that were available for this study at time 1 and 67 individuals at time 2. So we had these EVs, we DNase treated them, isolated the DNA, and performed qPCR using 4 different mitochondrial DNA primer sets. So what we first wanted to look at is what is the relationship between the mitochondrial DNA levels that were amplified from each of the 4 different primer sets. And so these, I know, are a lot of graphs, but I'll try to walk you through them in the sense of, so here's mitochondrial, the mitochondrial DNA levels that were amplified from 3164 compared to 4625. And what we found was that the levels of mitochondrial DNA were highly correlated and with each other in all 4 of the different primer sets when compared to each other. So there's a high correlation between the mitochondrial DNA primers. So what is the relationship between mitochondrial DNA levels and EV concentration? Remember previously I showed you that EV concentration decreased with age. So we wanted to ensure that if we saw differences in the abundance of mitochondrial DNA, that it's not simply due to differences in concentration. So we looked at the relationship between concentration and the mitochondrial DNA levels and found that there was no significant relationships with any of the four different primers and the concentration. So this so mitochondrial DNA levels do not correlate with EV concentration. So again, we wanted to look at how do mitochondrial DNA levels change with human age. And so this is linear mixed model regression, looking at the relationship between human age and the mitochondrial DNA levels from, so here's 3164 at visit 1 and visit 2, because remember we can do two different cross-sectional analysis. And so in all 8 of these comparisons where we look at mitochondrial DNA as a function of age, we see decreased mitochondrial DNA levels with age. And again, this is consistent in visit 1 and in visit 2 and in all 4 of the different primer sets. So this shows that EV mitochondrial DNA levels decrease with human age. And so again, these are in the cross-sectional studies. So what about longitudinal changes? So again, here we show a spaghetti plot where you have the different colors representing young, middle-aged, and old. And the start of the line is mitochondrial DNA level at time 1, and the end of the line is mitochondrial DNA level at time 2, and the length of the line is time. And a longitudinal analysis showed that there's decreased mitochondrial DNA levels over time. We can also perform linear mixed model regression in our longitudinal analysis as a function of looking at age and the mitochondrial DNA level. And our longitudinal analysis again shows that there's decreased mitochondrial DNA levels with age in all 4 of our different primer sets, using all 4 of our different primer sets. And since it's known that mitochondrial dysfunction contributes to aging and that EVs can be intercellular communicators. And so we wanted to test whether EVs isolated from young individuals or old individuals may affect mitochondrial function differently in cells, because as we've shown, EVs from old individuals contain lower levels of mitochondrial DNA. And so we, again, have EVs from our young individuals or old individuals, and we added them to cells in vitro and measured mitochondrial function using the seahorse method. And so we measured oxygen consumption rates as shown here in the graph. And here's the area under the curves, looking at both basal respiration and maximal respiration. So if we compare young cells treated with young EVs or PBS control, there were no significant differences in basal respiration or maximal respiration. However, if we compare cells treated with old EVs compared to young EVs, we see decreased maximal respiration and also decreased basal respiration in cells that were treated with old EVs. So this suggests that EV donor age can affect mitochondrial energetics in vitro. So just to summarize this part of my talk, what we found was that plasma EVs contain mitochondrial DNA, and with human aging, this mitochondrial DNA levels decrease. And just to bring it back to the first story that I told you, with human age, we find that there are changes in EV concentration, protein content, and mitochondrial DNA levels. And so as I mentioned, or Carolina mentioned, I'm a staff scientist in Dr. Michelle Evans' laboratory. She's the Deputy Scientific Director of the National Institute on Aging Intramural Research Program. So we have many collaborators within our institute, as shown here from LGG, LNS, LMBI, LCI, and TGB. And I'd also particularly like to thank some talented postbacks that we have had in the lab, Jamal Green in particular, and Stephanie Lazo, whose work I talked about today. They have both moved on to medical school. We have a talented statistician, Nicole Noren, that we work with. I'd also really like to thank the Handle staff and participants. We've collaborated on this work with Melina Jorgensen from Aalborg University and Ken Whitberg but we're at Johns Hopkins. And so I feel free to answer any questions now, or you can feel free to email me or find me on Twitter if you have additional questions after the talk. So I'd be happy to answer any questions now. Do you want me to stop sharing?
Thanks, Nikki. Yeah, well, if you want. That was great.
Okay. Thank you.
So we have a couple questions already coming in. Carolina, do you want to start us off? You're muted.
Yeah, just realized that. Yeah, so I'm wondering whether this mitochondrial DNA, do they actually have biological function? So that's my first question. And the second is it necessary to have mitochondria in the EV for the mitochondrial DNA to be transported in the EV?
Okay, so your first question. So it's thought that circulating cell-free mitochondrial DNA can act as a damage-associated molecular pattern, so a DAMP. So when you have damage to your cell or injury, the mitochondrial components can be released into the cytoplasm and extracellular space. And that the mitochondrial DNA, because it has very similarity, much similarity to bacterial DNA, it can then activate the innate immune system. And so then the activation of the innate immune system can then also cause chronic inflammation. And this may contribute, it's thought that it may contribute to various age-associated diseases, because it's known that as you age, you have increased chronic inflammation. Now here, what we don't know is that work is also plasma, thought to be more plasma free floating, where here we're looking more at the circulating cell-free mitochondrial DNA that's encapsulated in extracellular vesicles. So we don't know if there's a different role. And although, I'm showing that it's in EVs, there is still a portion that is in free floating plasma. So that I didn't have time to go in today, because I know sometimes with virtual seminars, I don't like to overload too much, because I think people get a little, Zoom overload. So I try to highlight, but that I did want to make that point.
Yeah. So do they actually intact mitochondria in the EV, or is it sort of like a fragment?
Yes. So others, not us, others have looked at electron microscopy and can see intact mitochondria, but there's also portions of mitochondria as well. So, it's still, I think, unknown in the field how much is really is it intact or is it fragmented? It's sometimes difficult to see, but some of the data does suggest that you can visualize mitochondria in EVs. And for your second question, it's, it's hard to do the experiment that you talked about, and we thought of doing it because there's some different cells that you can manipulate to get rid of their mitochondria. And so then you could look at whether mitochondria would then, if the impact of affecting function. This work started, right at the, or ended right at the end of the beginning of the pandemic. So those experiments would be really tough, but even tougher now in the pandemic.
So we have a question from Pule Rabani. Do you want to go ahead and ask your question, Pule?
Sorry, I forgot to allow. That was a really great talk. I'm curious, in that 5-year period, were any of the patients either going through pregnancy and then post pregnancy, related autophagy or weight gain or loss, and if that had any impact on their metabolism, and so the any kind of mitochondrial DNA impact in the EV?
So, we looked at, so pregnancy, I don't know actually that part. At the baseline of handles, we excluded anybody who was pregnant, they could not enter the study if they were pregnant. Now, whether anybody became pregnant over that time period, I've never checked that in particular, actually. Most people ask about age-associated diseases. So we have checked that. And so actually our work on diabetes actually began because of this study. So what we did was, it's hard in age-related studies to exclude anybody that has any sort of disease, because when you age, most people have something. And so in this study, our Vesicles with Age study, there were 12 people who were diabetic. And those individuals all had higher levels of EVs. And so that actually spurred us to look at EVs in the context of diabetes. But we did look at BMI in this study, and BMI was significantly correlated with EV concentration at time 2, but not time 1. But I think that was kind of driven because in this study, it wasn't designed to look at BMI. And so there was, for example, one person who was young, who had really high BMI. And so I think that made it kind of non-significant, for example. But in our further studies, when we looked at diabetes, it is an important factor to look at BMI. And so we usually try to control for BMI as well.
Thank you.
Okay, the next question is from Yunqi Chen.
Hi, hello. Thank you for a wonderful talk. I'm just curious about the aspect of agents. So during agents, the mitochondrial DNA content reduced in the EV, but also the monocyte activation by EV is increased. So is there any possible causal relationship with these two events?
Yeah, so we looked at that, and the problem is, it was in certain PBMC donors, but not in everybody. And, so I hated to we didn't make any strong conclusions about any of it, because we like to see if what we're seeing is consistent on all the different donors that we see, to make a real, significant effect. And, I didn't mention it, but our PBMC donors, we did do young and old at one point. And so it may be that it was in the old, but not the young, but those experiments are really hard to do, because to get the PBMC donors, you have to have somebody show up to the clinic for the, for freshly, apheresis unit, to get freshly isolated PBMCs. And you had to have a young and an old person show up at the same time. And we didn't feel like we could really cross compare all the different donors as far as ages went. So I don't feel like we have really good data on that or have shown it consistently. So.
Thank you. And just curious, what could be the possible PRR for mitochondrial DNA for innate activation?
Oh yeah, I mean, we haven't looked at that, but that's definitely, something that could be interesting. Yeah.
Okay, thank you so much.
Okay, so Sheila Abram has a question.
You're muted.
Yeah, you're muted, Sheila.
Yes, thank you. So thank you for your talk, Nikki. I was wondering, have you done any sort of quantitation in terms of mitochondrial DNA per vesicle?
Per vesicle? No, we didn't. And we get asked because people often will look at mitochondrial copies per se, like copies per mil, for example. And we decided not to use that approach because one, we looked at, when people do that, they usually have their one primer that they use, and then they do a plasmid control and then just normalize it to the plasmid control. And to do that, we decided instead that we wanted to look at different regions of the mitochondrial genome. And so we didn't normalize them to copies per cell, but it would be interesting because we can look at the concentration to look at, the different levels and compare that, per EV concentration, like what is the level, I guess you would say.
Yeah, because I think it's quite controversial in terms of like amount of DNA per vesicle, and does that really amount to anything physiologically relevant in terms of delivery? So I was curious about the mitochondrial DNA, because I would imagine that it would be less than total like nuclear DNA, no?
We didn't measure that. And I think sometimes, a lot of this depends on your like primer efficiency and, comparing nuclear to mitochondrial, because some people actually normalize to nuclear DNA with the mitochondrial DNA. Again, I don't feel like that was the best approach because in our hands, we were trying to amplify some of the normal controls that people use and didn't really see reliable signal in every single plasma EV preparation. And I think your comment about the levels, I think the levels do vary, because if you think about we didn't see a relationship between mitochondrial DNA levels and concentration. So I do think there may be differences. But to get to your question, whether it's functional, we do see different functional consequences in vitro. So for example, from old EVs, if you add them to cells, they they decrease basal and maximal respiration. Now, whether that's due to the mitochondrial DNA or it could be due to other cargo, that part we didn't obviously test in those experiments.
Yeah, that kind of brings me to a second part of my questions. So you saw a decrease of mitochondrial DNA as humans age, and you saw an increase of oxidative phosphorylation in your seahorse results. Is that correct?
Yeah. Okay. No. So I can't share, but anyway, so the oxygen consumption rate based on, so the basal respiration and maximal respiration were decreased in old EVs, in cells that were treated with old EVs compared to young EVs. So it's decreased.
But then, so is it equal amounts? Because you're, like, did you add equal amounts of EVs just because?
Yes, so we equalized the dose of EVs in that case, yeah. And we also did test the dose of EVs to see what dose we should use, because that assay is very finicky. And so I actually think when you have too many EVs in that assay, it actually clogs up the probe for measurement. So it's really important in all the seahorse assays, like to really do dose dependent for cells, for EVs, to really make sure your results are valid.
So just out of curiosity, how many EVs did you give per well? I guess it's a 96 well
yeah, so it was 8 times 10 to the 4th, so not many.
Oh, okay.
Yeah.
Okay, thank you.
So it's actually, I did the calculation because I thought somebody might ask this. So I think it was actually 2 EVs per cell. So it's not that many in that assay. However, like you don't know, a lot of these assays, like you plate a certain number of cells, they may grow, after you plate them. So, there's a number of those different factors, but in that assay, it's not many extracellular vesicles.
Okay, thank you.
Just to follow up quickly on that sort of theme about the biological activity of the mitochondrial or mitochondrial DNA containing vesicles, do you have any way of sort of, tracking those vesicles with some markers or purifying sort of enriched mitochondrial containing EVs that you could use to sort of test the effects on respiration?
Yeah, I mean, that would be, a really good idea. I mean, here we just used, we looked at in our data, EVs from young and old individuals and which ones had higher and lower levels. So, so we did sort based on that to use the pools of EVs from the donors that had different levels. So in that part we did, but we didn't track, we couldn't, like, for example, use an antibody against some of the mitochondrial components or something like that. We didn't necessarily do that.
Yeah, I would guess you'd need some kind of a surface marker or some kind of a subfractionation method or something. Yeah.
And if they're encapsulated, who knows if those markers are on the inside or outside. There's definitely mitochondrial proteins that people have found inside EVs as well. But that's technically most of them are intracellular. So I don't know if anybody's tested.
I think you can use MitoTracker to stain your mitochondria, and that easily passes through the cell membrane. So it should be a way to mark your mitochondrial DNA, your mitochondria.
Yes, that's true. Yeah, and that would be, it just would be, I think, if it would be bright enough to see, in your microscopy when you're doing your microscopy, I guess. Yeah.
Interesting. So we have a question from Lucia Languino.
Yes.
Hi,
Hi, Nikki. Great. Hi, Nikki. Super. Thank you very much for this lovely talk. I missed the first 5 minutes. You probably already answered, but because of the oxygen consumption and all this activity, and of course, an older person is gonna probably be less active than the younger person. Is that parameter taken into consideration when we compare the old and young EV generated from old or young people? older young people? And then the second one, is the concentration also measured with time during the day? If the circadian rhythm changing the amount of EVs, you know, we hear about that being also a potential problem in this kind of collection studies.
Okay, so we did not look at any sort of physical activity, you know, and compared that in the young and old in either of these assays, or, you know, when we set up this cohort. I'm trying to remember if we looked at any other parameter that would tell you that. You know, some of that data you have to really, I mean, we have, for example, in handles, we have a subset of individuals that we have accelerometer on. But you know, you have to be really good at some of the physical data when you collect it as far as their activity, you know, in questionnaires. But you can do some, and this is where we could look at some of the data. For example, we have measures, for example, of frailty, like that, you know, they do grip strength, you know, sit down in a chair. So there's a bunch of those physical parameters that we could definitely look at, you know, as it's known that exercise can increase EV concentration, you know, especially right after exercise. As far as the latter question, sorry, what was the latter question again?
During the day, if the collection.
Oh yeah. Okay, so all these individuals were the blood was taken in the morning and they were fasting. So just to exclude any of those possibilities.
Yeah,
we do that. Yeah.
Thank you, Nikki. In fact, I thought you would have said it. I missed that. I'm sorry. Did you get the what's the source of those vesicles? Do we know? Where are they come? Which cells release them?
Yes, so in this study, we haven't looked at the cell-specific extracellular vesicles in plasma. We did in the context of diabetes to look at different EVs that were in the blood and to find what EVs were different with diabetes. So we found that EVs derived from erythrocytes are higher in individuals with diabetes compared to non-diabetic. But here we didn't look at any of the cell type specific. That's not, I mean, that's something we would be happy to collaborate with, but even that assay we did, you know, the threshold, you know, you really have to be really good to make sure that you're not getting non-specific binding to the vesicles to really, and also if you want to do population percentages, you really have to do it in the same way, the same time. So, you know, that hadn't been our focus.
All right, thank you, Nikki. Okay.
Thanks.
Let's see, I don't see any other ones in the chat box. I guess I have one more question, which is related to, you had 46 proteins that you looked at for markers, and I was wondering sort of how you chose those. And whether you thought about, you know, doing proteomics or anything else.
So this is where it's great to be in a field that the technology is improving, because, you know, that was done, I thought about that, you know, because that was done 5 years ago, you know, and so much has changed, you know, and we initially did an EV array because that was really the best way to look at that time to do really, you know, broad, it was, I would even say it wasn't even an unbiased approach, because those were the ones that they had found that were present in EVs in plasma. And so we just kind of went with the best technology at the time, you know, to look at that. And the cytosolic proteins, we did take a more targeted approach, because when we lysed them in our mesoscale, again, the mesoscale kits even have expanded since then. But at the time, they had really good ones to look at apoptosis proteins, some signaling proteins, you know, various multiplex for signaling proteins. So that's at the time, you know, what we did. But I do think that that's really, you know, a good question. I mean, we've looked recently, we've used Olink proteomics to do some of their proximity extension assays on extracellular vesicles, and have gotten, you know, pretty good results given, you know, the multiplexing for inflammatory proteins, for example. And that's something we could certainly go back and do here.
Cool. Okay. Unless there are any other questions, I do just have one announcement to make, which is that the next speaker is going to be Christina Shu from Chile, and she's going to talk about honeybee EVs. Carolina, is that in the USA? So is that 2 weeks from now, or is that in Europe?
Yeah, that's the 2 weeks from now. That's correct, Alissa. So next week for the Europe region is Rosella Crescitelli from Sweden. So she recently published a paper in Nature Methods about EV isolation from tissue. So if you're interested to join that one, you're obviously welcome to join. It's just running at the time that probably most of the USA people are sleeping. Yeah, but we will have them on YouTube as well. So, and then the week after that is Christina Shu about honeybee EVs. I think that's going to be a very interesting talk as well. So yeah, thank you very much, Nikki. A wonderful talk.
Can I make another announcement? Carolina and I were talking about this. So Alissa and I are going to be part of this conference on February 25th and 26th. And Dolores and Jan Lotwell will be there. And the organizer told me that he has reduced the cost of participation. So he's offering for 4 people just the cost of one membership for $425 for 4 people. So I thought it was pretty good because I told him at ISEP we did it 100 and something, so it came down. So if you're interested, it's called Select Biosciences, and it's on the web. You can just look at under Select Biosciences. Okay. Sorry to interrupt.
That's all right. Yeah. Thank you so much, Nikki. Again,
all
wonderful
right,
talk.
Thank you guys for having me and for tuning in.
Yeah, and thank you so much, Alissa. That was great to lead the QA, and I hope you join us again next time to co-host.
I'll try. I'll look at my calendar.
Yeah. All right. Okay. Until 2 weeks then. See you then.
All right. Thank you, guys.
Bye.
Bye.
Bye.